Welcome, class, to the next lecture. As I always like to start out, we're going to start off with a dad joke. So, if a man is holding a bee, what is in his eye? It's beauty, because beauty is in the eye of the beholder. Beholder? Well, so what we're going to do is we're going to go over the different types of muscle and their structures and how it all plays a role with different contractions, allowing us to perform our bodily functions such as movement. We're going to go through each question. And so this first one says, describe the molecular characteristics of the contractile filaments found within the functional unit of the skeletal muscle cell, the sarcomere. So we're going to go dive into the details of it. But first, I'm just going to go a little bit larger and talk about how the sarcomere, aka the sarcoplasmic reticulum, the sarcomere, not the sarcoplasmic reticulum, is the whole cell, which, let me get my little pointer out. You can't really see here, but it's the whole cell, which is a little bit more in this green, so let me show a little bit. It's this whole green line right here. There we go. That goes down and across. And it has these T-tubules that go down it, so that way action potentials can propagate, open up some other channels, allowing calcium to enter into the cell. So that's where your sarcomere is located. We're going to go deeper and deeper into the cell. Within the sarcomere, you have your cytoplasm and also another space called your your sarcoplasmic reticulum, which is comparable to your smooth endoplasmic reticulum. In your sarcoplasmic reticulum, you have a higher concentration of calcium, which, if one of these channels over here, oh, my pen just died a little bit. If you open up one of these channels over here, this ryanodine, the calcium will flood into the cell due to the concentration gradient. Just going over some more structures and everything. Within the sarcoplasmic reticulum, you have where your motor units are going to kick in. One motor unit consists of multiple multiple units of the G-actin, F-actin, myosin heads, and a titin, along with the nebulin. So with this, nebulin is going to be on the Z disc, and it's just going to protrude from it in multiple spots on different areas. Titin's going to do the same thing. Titin is this black area right here, not the red. The red is the myosin. And then we're going to go over a little bit more that you can see in this picture. So this red here that you see all around, that is your myosin heads, and those will be able to attach to G-actin as long as the troponin moves the tropomyosin out of the way. These blue parts are troponin, and they are going to play a role in moving the tropomyosin out of the way, along with being able to have a couple other functions, which we'll get to in just a minute. But then this purple unit right here, which my pen died again, is the tropomyosin, which is blocking a binding site for the myosin. And this below the tropomyosin is your G-actin, which holds the right down here. It's all these circles. My pen just keeps dying over and over again. Well, we're going to turn on finger and use that. So just these little circles down here, not the blue ones, but the black ones. And that's your G-actin. Those are going to have little sites that allow you to bind to the myosin. So we're going to go over here and just talk a little bit more about G-actin. On the G-actin, you have those ATP binding sites. Whenever the ATP is removed from those binding sites by hydrolysis, which allows the site to be regulated and allow myosin to bind to it. Whenever you have the G-actin along with the tropomyosin and the troponin, that is going to be made up called the F-actin. And then tropomyosin, as we see, I just drew them like this, and then we have 3 different subunits of troponin. In troponin, you have subunit T, subunit I, and subunit C. The subunit T is going to be specifically bound to the tropomyosin. The troponin I is going to be specifically bound to G-actin, and the troponin C has 4 binding sites for calcium on it with 2 low affinities and 2 high affinities. This will allow you to perform the different functions that you need. Whenever you bind to the low affinities, these ones, that is what is going to allow the conformational change to take place of troponin to allow you to move the tropomyosin away from that myosin binding site, allowing you to perform a muscle contraction. On the myosin, this is a myosin type 2. There's 2 heavy chains. There's the tail, the hinge, and the head. You also have, and this red part that you're seeing here is the ATPase. And the purple parts are the light chains. And that's the alkali chain and the regulating chain that you have going on there. Next, we want to talk a little bit more in depth about this. So as you can see, we're going to describe the mechanism by which an action potential in the sarcolemma results in the release of calcium from the sarcoplasmic reticulum, followed by a description of the molecular mechanisms of muscle contraction. So we're going to slide on over here and just go a little bit more in detail about this, and I'm going to start from the beginning. So here you have your CNS, which consists of your brain and spinal cord. And just another fun fact for all of us to know is that we're born with all the muscle that we have. You can never increase the amount of muscle cells that you have in your body. You can only increase their size. But moving on from that fun fact, we're going to then move on to how the muscles receive their synapse. So the brain will then synapse down to the spinal cord into the PNS from the upper motor neuron to lower motor neurons. Then as it continues to synapse, it'll go to the peripheral nervous system, and it'll go through the somatic system, which then reaches the area in which the muscles are. And as you see here, these are multiple synapses that are occurring along each different muscle fiber. There's one there, one here, one here, another here, another here, another here, another here, another here. So they're just everywhere all over these muscle fibers because these motor units need to get the entire thing, all over these motor units, so that way it can cause all these muscle fibers to contract and perform their function. So I zoomed in this picture right here and just drew it a little bit bigger so that way we can focus on it. As that synapse comes across, it's going to be causing action potential down the neuron. As the action potential reaches the end of the neuron, it's going to open up voltage-gated calcium channels, as you can see right here. And that's going to allow calcium to flow into the axon terminal. Whenever the calcium flows into the axon terminal, it is then going to bind to the snare proteins. You have T snare proteins and you have V snare proteins, which is vesicle snare proteins and terminal snare proteins. Once the calcium binds to each, the snare proteins, the V and T snare proteins, which are going to bind to each other, allowing the vesicle, which is holding acetylcholine, to exocytose the acetylcholine into the so I just blanked for a second there. Into the synaptic cleft. And whenever it synapses into the synaptic cleft, that acetylcholine will then bind to nicotinic type 2 receptors, which are then going to it's going to allow in a cation, generally sodium, so we're just going to focus on sodium in this case. So we talked about these here, and there's your acetylcholine, and then it opened up a voltage-gated cation channel, allowing more positive to enter into the cell. As this happens, it's going to cause an action potential to sweep down the neuron, opening up voltage-gated sodium channels, which will then sweep down further. So I'm going to go a little bit deeper, but first I want to talk about this acetylcholinesterase over here. The acetylcholinesterase is just going to break down the acetylcholine, so that way you don't have a continuous contraction going on. And you also have nicotinic, these are nicotinic type 1 receptors, not nicotinic type 2. That was my bad. And then I show you the T-tubule over there, and that's where the synapse, the action potential is driving. As we drive down that T-tubule, you're going to reach DHP and ryanodine. That, which is, once the action potential reaches DHP, it's going to mechanically open ryanodine, allowing calcium to exit the sarcoplasmic reticulum into the cytoplasm. Once that calcium is into the cytoplasm, it is then going to cause a muscle contraction, in which I will describe a little bit more in detail here in just a little bit. Then I'm going to move over here. Once we have that calcium outside the cell, if we want to stop our muscles from contracting, we have to get that calcium out. So in order to do that, we're going to pump it back in through what we call the circuit pump. This uses a lot of ATP to pump that calcium back, and so part of the reason why it takes longer for your muscles to relax. Along these channels here, we also, just a quick reference to make sure that we understand our structures of the T-tubule, is you also have potassium channels that are allowing the flow of potassium out of the cell, and chloride channels flowing into the cell with those leak channels. The calcium is only going to flow from the sarcoplasmic reticulum into the cytoplasm. The reason why the calcium doesn't flow from the T-tubule into the cytoplasm is due to the concentration in the sarcoplasmic reticulum. The concentration of calcium is a lot higher, so therefore it is going to overcome the pushing of the opposite way of calcium trying to get in, which is why that is occurring. And so we'll talk about muscle twitches and twitch fibers here in a minute, but whenever there's a fast twitch, there's more circuit pumps working properly. Whenever you have a slow twitch, you have less circuit pumps working. And then next thing we're going to do is we're going to go through how that contraction happens over here. So this first one, I'm just going to explain what is on your myosin and talk about the actin a little bit. So over here, I'm going to just draw a little tropomyosin on there real quick, just so that way, well, that's with tropomyosin removed. Well, sorry, that's with tropomyosin, sorry. So with this first one, you have your myosin head, which is this piece right here. On it, you have ADP and PI. Oh, sorry, not inorganic phosphate. It's just a phosphate. Inorganic phosphate is free floating. But you have the ADP and phosphate. It will make a bent conformation, which will allow it to attach to the actin whenever the tropomyosin is moved out of the way. Whenever the calcium comes in, so when we move from here, that purple is the tropomyosin. Whenever we're going to move over and put calcium into the cell, that's going to remove the tropomyosin from the area, allowing the myosin head to then bind to the actin, which, as we know, whenever you are forming a bond, it is going to release heat. So that heat is released there, and it's also going, and whenever you break a bond, you require heat. So that heat is then going to go out of your into the surroundings, along with breaking the bonds of ADP and the phosphate. So whenever the power stroke occurs, the phosphate is going to fall off along with the ADP, which then your conformation is going to change from a 90-degree to around a 45-degree angle. So as you can see it moving over here to where it can pull that muscle fiber towards the other way. This is causing the Z disc to shorten, which I didn't talk about a whole ton earlier, but just something quick to mention is just going to pull those Z discs closer together, causing those sarcomeres to shorten in length. And then as that occurs, that ADP has fallen off, which means that there's nothing in that area anymore, allowing ATP to then come in. Let me just draw that in real quick. So ATP comes in and enters into that site. And whenever ATP does come in, it's going to form bonds, and so therefore it must release heat. And whenever that heat is released, it's going to go out into the surroundings along with help with the hydrolysis of itself. And so whenever that heat is released, it's going to break a bond and cause the muscle to relax back into the recovered state, allowing it to perform a muscle contraction again. So that starts with the recovery, and then you have the cross-bridge formation of the attaching to the actin. Then you have the power stroke where it moves. Then you have it releasing using the ATP, and then it recovers again. And we can repeat this process over and over until we have a force strong enough for us to do whatever function we are needing. Next, we're moving on where it says, in part 2 of this essay, discuss the various sources of energy utilized for muscle contraction. Include the relative amount of each source available to the cell, the advantages and the disadvantages of each type. So we're going to move over here to every source of ATP. First, I'm going to talk about each one, and then we're going to compare and contrast them a little bit and the advantages. So first we have cytoplasmic stores. This is available instantly, which is very nice. We can access it very quick, but it gets depleted very quick. As you can see, I wrote here. Oh, let me switch to my other pen. Let me see what this does. Nope, don't want to do that one. But yeah, as you can see here, as I drew out there, its availability is limited, so that means you can run out pretty quick, which is part of the reason why it only lasts 1 to 5 seconds. So it's very convenient, but it also isn't a long-term process that you want to keep using over and over again, because you won't be able to keep it up. And this is used at the start of a muscle contraction. The creatine phosphate, that combines the ADP and an inorganic phosphate, or a creatine phosphate in this case, in order to make ATP, which can then be used as the energy that we need. Creatine phosphate usually lasts about 10 seconds for these contractions to occur. Its availability is limited, but it does have a rapid regeneration, but it does have a short duration. So that means you can do short bursts of high intensity activity, such as like sprinting, weightlifting, or doing things that you would normally have to require a lot of energy in a short amount of time. Next one up, we have glycolysis. This one is the breakdown of glucose into pyruvate, which produces ATP without oxygen. This one generally helps you maintain the muscle contraction and different things for up to 60 seconds. Its availability is moderate. It's in between the stores in the creatine phosphate and aerobic metabolism, which is the highest. The advantage is that it has quick ATP production, so that way we can do different things. One of the disadvantages is it produces lactic acid, which then just gets stored later, and so that way it's not like the continual, so it's just producing the lactic acid, causing your body to have more molecules in the body, which then have to be transferred elsewhere. It is generally used for moderate to high activity lasting 30 seconds to 2 minutes. So these are some different things that like your mid-distance runners would say, or like the people who just go to the gym for like who try to do it like a 30-minute workout, something that where they can be consistent but don't wear themselves out by the end of their exercise. The aerobic metabolism that uses oxygen to break down glucose, fatty acids, or amino acids in the mitochondria, and this is high in availability. This is the one that produces most ATP. You're going to see this in your Ironman people. You're going to see this in your marathoners, whenever you jog, whenever you cycle, doing things like that, because it produces the most ATP and it has high availability. So that way you can access it and keep using it over and over and over. The issue with this, though, is that it's slower to activate. So as great it is, it's just not the best in the being a quick source. So I made a little diagram here that talks about the speed, duration, oxygen, ATP, and what it's used during. So as you see, the cytoplasmic stores are immediate. They're 2 to 3 seconds. No oxygen is required. 1 ATP is yielded, and it's the start of a contraction. The creatine phosphate is very fast. It's around 10 seconds. No oxygen required. 1 ATP, and it has short, intense bursts. The anaerobic glycolysis is fast, 30 seconds to 2 minutes. It doesn't require oxygen. It yields 2 ATP, and it has moderate to high intensity, and it's short term. Aerobic is slow in speed. Duration lasts hours, and the oxygen is required along with yielding 30 to 36 ATP with long duration and endurance activities, which allow you to do those different types of things. So moving on to our next topic, we're going to describe the two main types of smooth muscle, describe the molecular mechanisms of muscle contraction in smooth muscle, beginning at the point of calcium entry and at the point the contraction ends. So I drew out this little diagram again, except in accordance to how smooth muscle works. So first we got to understand that there's two different types of smooth muscle contractions. There's phasic contraction and the tonic contraction. Each are different based on, in accordance to make Brother Hunt happy, calcium. But I still like to say it's due to the MLCP. But to make Brother Hunt happy, I'll say calcium. And so with this being said, it's all about when that MLCP occurs. Is it going to occur late or is it going to occur early? If it occurs late, it is going to be phasic or phasis. And with this, the exact same kind of thing is happening, such as the skeletal muscle, but just a little different. Whenever you move from your recovered state or your beginning state to attach to the actin, whenever the tropomyosin is removed, you have your MLCK, which is your myosin light chain kinase, meaning it's going to add a phosphate on there from using ATP. It's going to take some myosin take a phosphate from the ATP and place it on that myosin chain, which pushes tropomyosin away. And and the calcium, of course, is activating this. Which, once that occurs, your phosphate and your ADP are going to fall off, allowing that power stroke to occur. And then you still have that second phosphate on there. ATP will then come in, causing the release from the actin head. And then as long as you have that phosphate on there, you can go right ahead into another contraction, just over and over and over again. But if MLCP comes in, which is myosin light chain phosphatase, it's going to remove that phosphate, causing it to go into its original state, into the recovered state, allowing it to have to restart the cycle all over again. So that's your phasic pathway. And then we also have a tonic pathway. The tonic pathway is similar, but the MLCP, the myosin light chain phosphatase, comes in early. So rather than it coming on that last part, it's going to come in on the second part, as we see right there. So we have an unattached ADP and phosphate, and then that calcium is going to come in and help remove the tropomyosin. The MLCK and ATP are going to come in and add that phosphate on the myosin. Then our MLCP, after the phosphate and ADP come off, the MLCP is going to come in and remove that phosphate, so that way the conformation changed a second time, allowing it to return to that formation. Whenever that conformation changed a second time, the ATP is going to come in, and that ATP is going to try to hydrolyze, but due to that conformation changing a second time whenever that phosphate was removed, that ATP is going to have a harder time coming in, so it's going to take a lot longer because it doesn't want to release. So we call this the latch state, as it takes a long time in order for that muscle to reach that relaxed state, that recovered state. So I drew a little diagram down here for us, just talking about our activity. So over time, the enzyme activity is going to be different for each one, such as MLCP, MLCK, and calcium. The MLCK is going to be constant like a linear line. The MLCP is going to look a little bit more like an action potential graph, along with the calcium, and they're going to follow along accordingly. So you're going to have high activity, start with low activity, then you're going to move up to high activity, and then you're going to move back down to low. While with the tonic stage, you're going to have a linear activity rate for each MLCK, MLCP, and calcium until you start to plateau off a little bit more. Also, we see here that your calcium, this is what happens when your calcium levels are increased or decreased too. Say calcium levels are going to increase over here and calcium levels are going to decrease over there. And just to show you a little bit about how MLCK works, is this the inactive state of MLCK, but then calmodulin will come in, which has calcium binding sites, and that's going to bind to the MLCK, causing it to be in its active state. And the MLCP down here is going to be inactivated whenever you have a phosphate on it, but then rho kinase comes in, and rho kinase will want to come in. But whenever you have sustained calcium, it's going to inactivate the rho kinase, and then the MLCP will no longer have that phosphate on it, which will then cause it to be active. And we just have down here a little bit about our different channels, such as voltage-gated calcium channels on the cell, stretch-activated calcium channels, and ligand-gated calcium channels, which just are different ways for a tonic stage to occur, tonic contraction to occur in smooth muscle, because it's moving, the calcium's moving from the outside to the inside, while for phasic, the calcium is going to move from somewhere inside the cell, deeper inside, to somewhere inner inside, such as the cytoplasm. So we see that happening there. And then next we're going to move on to our next question, where it says, compare and contrast smooth and skeletal muscle structurally, functionally, and regulation. First, I'm going to talk a little bit about our muscle fibers a little bit. So these are generally found in skeletal muscle. Your type 1 muscle fibers are going to be slow. The circuit pumps are going to be slow. You're going to have troponin. They're going to be, the vessel sizes are going to be small in diameter, which means that different non-small, non-polar molecules can move across. Your myoglobin is going to be high, capillary density high, resistant. It's going to be resistant to fatigue, but it's going to have a low power output. Oftentimes the people who have more of these are generally your distance runners, people who do really good running long distances. Those are your marathoners, your joggers, wherever you go. Then you have type 2A muscle fibers, which those you see about less than 1% in humans, but you see the majority of them in animals. This one, your circuit pump is going to act fast. You have troponin. They're going to be medium in diameter. The myoglobin is going to be high. Capillary density is medium, but this is where it comes in handy. So the power output is medium. So not super big, not super low, but it's going to be resistant to fatigue, meaning it can last a longer period of time, allowing it to keep performing. Then you have your type 2X muscle fibers, which you also see in humans, a bit in humans. These are going to be you guys who are like power lifters, like you see a lot more in power lifters and things like that, who are really taking advantage of this. Your circuit pumps are working fast. You have troponin. They're large in diameter, which causes a little bit of diffusion issues because it's larger, it has to cross a bigger area. You have low myoglobin, low capillary density, and they're more easily fatigued, but they give off a huge power output, which allows the things to give you a lot more strength whenever you perform an exercise or something like that. And then another thing that I just want to summarize is skeletal muscle is voluntary, it's striated, and then it is for quick, forceful movements. So this allows you to do actions for a short period of time, allowing you to get a job done that may need to be done. Your smooth muscle is going to be involuntary. It's non-striated. And these are your slow and sustained contractions, because that'd be really awkward if your smooth muscle was not slow and did not have slow contractions, because you don't want your smooth muscle running out, otherwise you'd be pooping your pants all the time. That sphincter would not be working right, so you definitely don't want to embarrass yourself in public. It's not fun. And so that's the slow, sustained contractions. And just going a little bit more into the differences of each of these. So some of the shape differences is that the skeletal muscle is long and cylindrical and multinucleated, while the smooth muscle is a single nucleus, mononucleated. The arrangement of skeletal muscle is in fascicles. I wish I had my pen working better. Let's see if I can actually plug this in real fast. Maybe I can get this working while I'm doing this. There we go. Okay. Now I'll start being able to write prettier. So these are going to be your fascicles. And that's how they're arranged. While you're, let me turn my finger off now. While your smooth muscles are going to be arranged in sheets or layers. Along with their functional differences is that we talked a little bit about, and the skeletal muscle can fatigue with prolonged use, while the smooth muscle takes longer to fatigue. And the skeletal muscle is very much for movement, posture, like producing heat, things like that, while your smooth muscle is going to be for the moving of substances. Just throughout your body. Next up, we're going to focus on our next question here, which is finally describe and explain and then compare and contrast the muscle sensory receptors, muscle spindles, and Golgi tendon organs. Describe the stretch and reflex and the withdrawal reflex. So I'm just going to go over this really brief. We have upper motor neurons and lower motor neurons, which the upper motor neurons will go synapse with the lower motor neurons, and the lower motor neurons will then leave the spinal cord and then synapse with skeletal muscles of the neuron. And these will cause different reactions. Through our different receptors, we have mechanoreceptors, which we feel mechanical things such as touch, pokes, like things that things that we can feel on our skin. Then we have chemoreceptors, which we can then which is your chemical stimulus. Thermoreceptors, temperature stimulus. Photoreceptors, that's your stimulus to light, gives you vision. Nociceptors, those are like your pain receptors. They respond to chemicals released from, and they can indirectly respond to chemicals released from damaged tissue. So, talk very quickly about muscle spindle and Golgi tendon. We're going to get there in a second. We're going to first, this was the first one. To talk a little bit about your muscle spindle and your Golgi tendons, muscle spindles are embedded within skeletal muscles. They detect changes in muscle length and rate of stretch. They're intrafusal muscle fibers surrounded by sensory nerve endings. And when the muscle is stretched, muscle spindles activate and signals the spinal cord to initiate a reflex contraction of the same muscle, which allows that stretch reflex to come in, which we'll talk about here in just a minute. And then it helps maintain muscle tone and posture, as we see that skeletal muscle does. With the Golgi tendon organs, those ones are found on the junction between muscle fibers and tendons. They detect muscle tension, such as the force of the contraction. They're encapsulated nerve endings interwoven with collagen fibers. And when the tension is high, Golgi tendon organs, they send inhibitory signals to relax the muscle to prevent damage from overstretching, causing those Z discs to be too far away from each other, which is what that overstretching is when those Z discs are too far away. And that's the inverse myotic reflex. And these help protect muscles and tendons from excessive force. So, just comparing real quick, muscle spindle detects muscle stretch located within the muscle belly, like inside the muscle. Its response causes muscle contraction, and its function is maintain posture and tone. While the Golgi tendon organs it detects muscle tension. It's at the muscle tendon junction. It causes muscle relaxation and prevents muscle damage from overexertion. Finally, to talk on our last subject, we're going to talk about our different reflexes. We have our stretch reflex and our withdrawal reflex. The stretch reflex is a sudden stretch of muscle. Responds to a sudden stretch of muscle. That's the stimulus. And its receptor is the muscle spindle, and it's monosynaptic. Only one synapse is happening between there. And then the response is the contraction of the stretched muscle, such as the patellar reflex, the knee jerk, whenever the doctor hits your knee and your knee kicks a little bit. Its purpose is to maintain posture and tone, which, look at that, goes great with the muscle spindle. Then we have our withdrawal reflex. That stimulus is painful, is whenever it responds to pain or noxious stimulus, such as your chemoreceptor and your nociceptors are going to come and clutch here. The receptor is your nociceptor, and its pathway is polysynaptic, which involves interneurons. It's going to have multiple synapses. So the response is the withdrawal of the affected limb by contracting flexor muscles. And an example for this is just pulling your hand away from a hot surface, pulling your hand away from being poked by a needle, or stepping on something sharp or for all those dads in the room, stepping on Legos, and they just protect the body from harm. And here's a comparison, a little bit just brought up in a little chart. Your stimulus for the stretch reflex is muscle stretch, receptors muscle spindle, synapses is monosynaptic, your response is muscle contraction, and function is the posture and tone. While withdrawal reflex, it's the painful stimulus, its receptor is nociceptor, it's polysynaptic, it causes you to withdraw your limbs, and it protects you from injury. Well, I'm glad you had a great time in class today. Hope you enjoyed the rest of your classes and the rest of your videos that you have to watch. Make sure to tune in for next time, so that way you can always know how your body works. Thank you.